Hello, everyone. My name is Kaan Salcin, and I will be talking about my research focusing on PET imaging of epigenetic processes in human stimulant drug users. According to the National Institute of Drug Abuse, drug addiction is one of the leading causes of disability in the world. In Canada alone, the non-medical use of drugs produces an annual economic cost of around $38 billion per year. Among illegal drugs, a disproportionate burden is attributed to cocaine and other stimulant drugs like amphetamine and methamphetamine. A recent epidemiological study from last year actually revealed that the estimated cumulative conditional probability of substance use disorder was highest for stimulant drug users, with around 50.4%. Yet little is known about factors that predict the likelihood of this transition from use to disorder. We have very strong evidence from twin and family studies that inherited genetic factors play a substantial role in the development of maladaptive drug use patterns. However, adoption studies underline that the environmental factors, especially aspects of familial dysfunction, also strongly correlate with substance use disorder risk. So these aspects include childhood maltreatment, supervisory neglect, parental loss, and a lack of protective adult relationships, which we will be collectively referring to as early life adversity, or ELA. Studies in laboratory animals have identified a number of epigenetic processes, such as DNA methylation or histone modifications, histone acetylation in the brain, as promising candidate mechanisms through which early life adversity and initial substance use itself actually can alter gene expression patterns and brain responses to rewards and punishment. And these enduring alterations can shape personality traits and influence susceptibility to substance use disorders. Based on recent animal data, one class of epigenetic enzymes, histone deacetylases or HDACs, might be of particular interest. In rodents, exposure to ELA, or early life adversity in the form of maternal separation usually, alters HDAC activity in certain brains, and similarly, pharmacological and viral modifications of class 1 HDACs result in changed behavioral responses to subsequent drug use. Now, my PhD project aims to measure these processes in the living human brain using the first ever validated in vivo epigenetic imaging tool, the positron emission tomography, PET, with 11C martinostat. So based on studies in laboratory animals, I hypothesized that in humans, early life adversity severity, as measured by a structured clinical interview and pertinent questionnaires, will alter 11C martinostat binding, which reflects HDAC availability in different regions of the brain, in the cortex, hippocampus, and parts of the striatum. And this variation, in return, will be correlated with differences in lifetime substance use measures, as well as differences in mood and personality traits like impulsivity or sensation seeking, and reward learning and cognitive function measures quantified using a variety of computerized tasks. We are currently in the arduous process of recruiting participants into 3 experimental groups: stimulant drug naive healthy volunteers, stimulant drug users without a substance use disorder, and individuals with a stimulant use disorder. And mediation analysis will test whether differences in HDAC expression mediate the effect of ELA and early drug use on various behavioral outcomes and neurobiological features. So far, 20 participants have completed the study entirely, and preliminary analyses are underway. Here are several normalized PET images from a selection of our participants, but we're getting there. And by benefiting from the recent development of tools, this study has the potential to improve our understanding of the link between early life experiences, epigenetic modifications, and enduring susceptibility to addictions in humans. And by coupling these results with behavioral, neurobiological, and gene editing studies in laboratory animals, we will have an unprecedented opportunity to characterize drug use-related molecular pathways to addiction. Thank you.